everyone, my name is Nadine Schneider and I'm a data scientist at Novartis. I would like to thank the organizers for inviting me to present at this workshop here. It is really my pleasure today to give you an industry perspective on the application of machine learning in drug discovery. I would like to start with a little bit of introduction to our field to set the stage. Over the past years, we have seen that the digital revolution finally reached the pharmaceutical industry and that machine learning models are becoming more and more relevant for drug discovery. One indicator of this is, for example, on the left plot, the number of emerging AI startups tackling drug discovery. And on the right plot, you see the number of collaborations between AI startups and big pharma. The second indicator of the enormous interest of AI for drug discovery is also mirrored in the exponential growth of publications in that field. On the right side, you can see a plot showing the papers that have appeared every year, starting in the 1970s with single publications and reaching now more than 1,500 papers per year. This means you would need to read about 4 papers every day to keep up with the development in that field. And this only captures the articles published in peer-reviewed journals, not to talk about preprints. Now let's talk a little bit about the drug discovery pipeline. This is a very long and very expensive process where most of the programs will not result in successfully bringing a new drug to the market. In early drug discovery, usually the target causing the disease is identified and promising drug candidates are developed. In the preclinical phase, then the drug candidates are further profiled in vitro and in animal models and tested for efficacy and safety. 1 to 2 successful drug candidates then enter the clinical phases, and they are administered the first time to healthy humans and later patients. Only if all of these phases are successful, the drug candidate is officially approved by agencies like the FDA to be a drug. On average, this takes about 10 to 12 years and costs up to $2 billion. Especially in the last 2 years, more and more of the research in our field appeared in articles for the general audience. Promising headlines like startup used AI to design a drug in 21 days or AI model yields new drug to overcome antibiotic resistance are showing the hope that people have in applying AI to drug discovery. It would be really wonderful if it would be that simple, but please remember the definition of a real drug I gave you on the slide before. So there's still a long way to go and we should be careful with these announcements. One last point I would like to mention before I will talk about our projects around machine learning and early drug discovery in Novartis is the long history of computational prediction methods and the early adoption of machine learning in cheminformatics and computer-aided drug design. So this really dates back to the 60s when QSAR, which stands for Quantitative Structure-Activity Relationship, was invented. Until then, we see a continuous development of novel models for the design of new molecules or molecular representations. Also, the first neural nets were applied to chemistry problems in the 90s. Okay, now let's start with a deep dive into our experience and projects around machine learning for molecules in Novartis. This slide gives you an overview about the topics I would like to touch on in the next 15 minutes. We will talk about what is important and how do things work related to data, productizing machine learning models, including users, and also novel models we are developing. The size of the boxes give you a rough estimate of the importance we have experienced for these different topics in the past years. First, let's talk about data. Often the perception from the outside world, especially coming from academia, is that pharma has this massive treasure of data. Yes, it is true, we are generating massive amounts of data in certain areas, but usually when we then go into the details, things become much more complicated. So I would like to give you some insights on how the data looks like we usually deal with in chemistry. On the right side, you can see a rough summary of the challenges we have with our data, and I will go into more details on the next slide. So one point I would like to highlight here is the generation of the most relevant data is the most expensive. So for example, in vivo data, but also data that requires the synthesis of novel molecules. Okay, let's talk about big data first. So we have roughly 14,000 in-house data sets measured with measured biological activity against different biological targets. We call them assays. This sounds not too bad in the beginning, but when we then dive deeper into this, we find that 50% of these data sets contain only up to 500 measurements, so not very big. Only 2% of these 14,000 assays have more than 10,000 measurements, which are in the range where novel deep neural nets can be applied more reliably. Now we could think about using multitask to model this big matrix where we have many compounds and measurements in different assays. But when we look at the completeness of the matrix, we find that it is only 0.1% complete. Another challenge is that for about 40% of this data, we don't have a quantitative measurement, but only a qualitative value. So you can see going into the details with that data, it becomes quite challenging. Next, let's talk about the data distribution and the similarity principle, which is important for machine learning. Here I would like to mention the QSAR concept that I earlier introduced and that is established for a long time in our field. It means that a similar chemical structure most likely will have a similar biological activity or physical property. So, however, since a while now we know that so-called activity cliffs exist in our data. This means that even tiny changes in the molecule can lead to strong changes in the biological activity. For example, on the right side, you can see a so-called chiral cliff. Here only the change in the three-dimensional constitution of a molecule leads to a strong change in its biological activity. So these cliffs are very hard to model with a machine learning method as they really represent a discontinuity in the data. Okay, going back to big data again, you might all have heard that the chemical space for drug-like molecules is super large, estimated to be in the range of 10 to the power of 60. But how much do we actually know about this space? Recently, some super big virtual spaces or on-demand accessible spaces have been created. But when we now look what has been synthesized already, we find that only 169 million molecules are in cars. So there's a clear gap of what has been made and what could be made. Linking this to the measured biological activity, we find, for example, in PubChem, there are 272 million measurements. So this sounds pretty big, but the data is highly biased and clustered because until today, it is still humans that select what to measure and what to make next. This is often a compromise between the interest and the chemical feasibility. So when you now look at different pharma companies and different projects, you will find that certain areas or islands of the chemical space are strongly exploited, while many other areas are not explored at all. Due to this, we have a very biased sampling of the chemical space, which makes it very hard to create models that are generally applicable. Finally, the last point I want to mention on our data is that it is strongly evolving over time. To make this more clear, I would like to show you an example of an in-house protease dataset. So on the x-axis, you can see the evolution of this assay over time, and on the y-axis, you can see the number of measurements. Biological active compounds are colored in blue, inactive ones are colored in green. In the beginning of the assay, you can recognize that chemists were quite successful in creating active molecules. At some point in 2009, no actives can be found anymore. So why did this happen? So after some investigation of this dataset, we found that the assay was not used for a specific protease project anymore, but that it was used as a panel assay to test for off-target activity. So in that case, you hope your compounds are inactive in that assay. So without a deeper dive into this data and understanding how medchem projects work, you will most likely result with models that are not successful. Okay, the next part in my talk is about productizing machine learning models. This is an extremely important point for us, as we must ensure in this highly regulated industry that our models are reproducible. The models are used to help really with important decisions, so they need to be reliable. For this, we have, for example, a board of experts that reviews each model before releasing it to the research community. And the most important point is we really need to democratize our models so that all scientists can benefit from them in their daily work. Okay, here I want to share with you a very nice illustration that Scully and team at Google published a few years back. And it is really well captured and eye-opening when we think about productizing machine learning models. Often people talk a lot about their new innovative machine learning models, which is super important scientifically, but unfortunately it only constitutes a small piece of a productive machine learning model. So here we need much more. We need easy ways to collect, verify, and prepare our data. We need to make the model scalable and robust. We need to ensure that the model is correctly configured and that feature generation is coupled to the model at prediction time. So we need to monitor its performance over time, and we also need to allow it to be integrated in analysis tools. So there are many more pieces at that end that make a machine learning model truly actionable. To help with these challenges, we have built a predictive learning environment in-house, which is called Plenty. It uses a containerized software environment that ensures the reproducible data science. We have implemented it as a Python-based software framework that provides abstraction layers and interfaces between data handling and machine learning libraries. We also provide a collection of codified best practices to help with creating and using predictive models. Finally, and most important, it builds the foundation for model deployment, for our model deployment infrastructure to make the models broadly available. Now, before productizing a machine learning model, we want to have a defined way to select the best model for our use case. For this reason, we have created another framework that helps standardized model building and benchmarking. Here also a less experienced data scientist is able to input a dataset consisting of molecules and experimental measurements and to define a list of models and molecular representations that should be evaluated. The data scientist can then select the best model and further prepare and deploy it with Plenty. Another way to democratize machine learning models is shown here. So we are building an automated pipeline that pulls in the newest MedChem project data and creates a set of models given different chemical theories and assays. The results are then visually prepared and pushed back to the analysis platform that chemists are using in their daily work. If they see a model that is reliable and useful for their current project, they can reach out to a data scientist who can further refine the model and deploy it using Plenty. Okay, the next part in my talk are the users. So to make our machine learning models really effective and actionable, they need to be accessible and integrated into the daily work of the chemist. But it is not enough to technically enable them, but also to change their thinking and to gain their trust in the models we build. We try to enable this by investing in data preparation and consolidation and work on comprehensible result reporting. An important aspect is also the education of the users to deepen their knowledge and create the right expectations on our machine learning models. So we invested much in data preparation and consolidation over the past 2 years to facilitate really the extremely complex work and decisions medicinal chemists are facing in their projects. The data that comes together in a drug discovery project is really overwhelming, not only the amount of data, but also the complexity. So the project teams always need to be on top of that data to decide which experiment to do next. On the left side, you can see the numerous sources of data and many different data types that are needed and need to be integrated and connected to help with the analysis and decision making in a drug discovery project. For this reason, a large team of data scientists and engineers work together with business experts to create IGPS, an interactive dashboard for MedChem projects. It allows project teams to easily set up different views for their project to analyze, for example, toxicology data, look at assay correlations, monitor the performance of our machine learning models in respect to their project, and to get for each molecule all necessary information in a very convenient way. And this is truly changing how project teams work and how they use our machine learning models. Let me show this to you on a more concrete example on the next slide. Okay, having easily access to IGPS on model predictions, the slide shows how our solubility model was used in one of our MedCom projects. On the left side, you can see the predictions. On the x-axis and on the y-axis, you find the number of measured compounds. The bars are colored in the measurement value. So you can see that the model predictions are quite reliable. In IGPS, you can now track this down to each series in a project. On the bottom left, you can see for one series of the project, the model was not able to give reliable predictions. This means that for the team, they need to go and get measurements for new compounds in that series. Now talking about cultural change. So on the right side, you can see the evolution over time of compounds they sent to the solubility assay. With the introduction of IGPS and the so-called digital lead in the project, who is helping with the analysis of the data and also with the digital education of the rest of the team, we see a clear change in behavior. First, fewer compounds are submitted for measurement because the solubility model gives reliable predictions for that project. And second, the overall quality of the compounds submitted for measurement has changed to a higher fraction of highly soluble compounds. So this truly shows how impactful these models can be if they are accessible to the chemist. Last but not least, our novel models. We are engaged in several projects where we try to establish and implement novel machine learning techniques for the really challenging problems. And let me quickly introduce two of them on the last slide. So one project where we apply and develop active learning methods is the microcycle platform. Here we are really trying to close the loop in the design, make, test, analysis cycle. It is a fully automated platform to design, create, and profile novel compounds using microscale blade-based synthesis. In this platform, machine learning is driving the design of the chemical libraries. The results of the different assays then are fed back into the machine learning models for the next iteration. It is also very interesting to mention that the data capturing and handling really has driven the setup and the automation of this platform. Another project currently is building an integrated de novo design platform where the goal is really to inspire medchem teams with high quality compounds that fit their target property profile for the project. This is in collaboration with Microsoft Research in order to bring together the experts in data science with the experts in drug discovery. We are implementing here a set of diverse generative models coupled with multi-objective optimization methods that then make use of the property prediction models I introduced to you before. And with that, I would like to end with some take-home messages. So most important in our domain is that you either have gained some domain expertise already, or that you team up with a domain expert to be able to create useful models for drug discovery. It is incredibly important to understand our data before really starting to model it with the newest methods. At the end, what will really create impact are actionable and reliable models that can be easily accessed by our medchem teams. I hope now I could give you some more insights into our domain, and I hope you are excited now to come and help us with the journey of digitalizing drug discovery. I would like to thank all of the people that are involved and have contributed to these different projects and initiatives I presented to you. And I want to thank you for listening. Thank you.
Fantastic. Thank you, Nadine, for your very insightful talk. And we're going to take questions for Nadine here on Zoom or in Rocket Chat on the NeurIPS website or on the Discord channel. And the first questions are coming in. I'll start to read them out. So the first question was, Nadine, if you could give more details in your education program for chemists.
Sure. Can you hear me?
Yes.
Hello, everyone. Yes, so the education program for the chemists, actually this started as an initiative. I think we started last year, but this year was really then a big year for that, because many of the people had actually to work from home, right? So they had time to learn new things. So we had regular meetings with them. It is a part of our computer-aided drug design group that are very involved in this, but also other people in the company that work on data science projects that then join regular talks, update people on what is there, and really train them also to use the methods, but also how these models can be interpreted, what it means, how is the reliability of the models. And yeah, we continue on that because I really think it's a good way to broaden the knowledge of our scientists and make them aware of the new technologies that are out there.
Great, thank you. And then the next question is, what does the expert review of models before deployment involve at NIBA beyond looking at quantitative metrics?
Yeah, I think mainly it is really looking at quantitative metrics. So what we usually do when we deploy these methods is really preparing a slide deck and some dashboards with all of the model results on our data. We usually do time splits, so to get some prospective results for the models. Also look at the performance in different projects, not overall, to see how good they perform. And then there's a discussion ongoing if all of the criteria are met, so if the reliability is high enough overall for the models, and only if this board of experts agrees on that the model is good to hand it over to the scientific community, then we deploy these models.
Great, thank you. Then the next one. You mentioned different data sets. I imagine that many of them are internal to Novartis. Are the public data sets sufficient, according to you, for academics to develop useful new ML methods for drug discovery?
It's a good question. Yeah, I think most of the data we use for sure is in-house data, but we also, for example, training these generative models, we make use of public data as well, right? So it really depends. I think the really, let's say, important models for the project teams are the ones that have the most difficult and most expensive data. So when it comes into in vivo studies and tox studies, and even with this data, which is very limited also in the company, and which is very, let's say, scattered over the chemical space, it is we cannot build models with them at the moment. So it is too little and too biased to do that. And for the other data, so like solubility, permeability, and so on, we use our in-house data. And I guess there's also only limited data available in the outside for really academia to try this out, which is sad, I must say.
But then maybe to follow up on that, do you think then still the lessons you would learn from, let's say, developing on publicly available data can be transferred to some extent to the in-house data, or is there no transfer possible at all?
It is difficult, I would say. It really depends. So, for example, let's say log-P, this is a super simple data set. I think there's no problem to use public data because it's very similar to what we measure in-house. But for example, solubility, where you have a data set in the public, which is not too big, and it spreads over several log units, this is not really comparable to the data we have in-house. which has a much smaller range and it is less well distributed, I would say. Yeah, I think what should happen more is collaborations between academia and industry, so that we can test the new model methods on these data and also give insights to the people outside.
Great, thank you. Next question. The graph illustrating the effect of introducing IGPS and a digital lead on compounds submitted to the solubility assay was impressive. Are you worried that this will result in the collection of imbalanced data going forward, i.e. you'll end up collecting not as many examples of insoluble compounds? Any plans compensating for this?
Also a very good question. I think we are aware of that. It is not that we see that right now, but yes, we kind of bias our data sets even more if only, let's say, the super difficult compounds are measured. So we are also looking more and more into active learning so that we can measure compounds that give us more information, right? So that it's not always the stuff that people select which are important for the project, but maybe also things that would enhance our models for future use.
Great, then I think one more. What role does explanation and interpretation of models play for model review and uptake in your end users?
It is at the moment not super important, I would say, not for these large-scale models. I mean, people are always curious to know what are the reasons why a model makes a prediction, right? But I think at some point, many people also accept if the performance of the model is good enough that they are okay with just the number. But if we could get more insights here, I think especially for the data science community, this would help a lot also to improve models.
And maybe one quick one. Thanks for the talk. You mentioned you have a fully automated DMT, so design, make, test loop. Is there any backlash or competitiveness from chemists if they are left out of this loop?
I think what we try in all of these initiatives I showed you is really not to leave out any people. So it is really to make collaborations between the different groups. And I think we can never do this without the knowledge of the chemists. So there's a strong collaboration in that loop. So it is actually that the chemistry teams come, submit their proposals first. So tell us where the limitations of the series is and what they want to improve. And then I think the cycle starts, and also they get results in between, and we can change direction if necessary, because nothing is perfect at the moment, right? So, and we cannot run all of the reactions at the moment, so there's also room for improvement here.